Okay, so in this presentation, our group has gone through and prepared a presentation using the Tuxedo Suite to analyze RNA-seq data using Galaxy. And so what we've got here is, just to begin with, we want to go over what RNA-seq is. RNA-seq, as many of us know, is a technology based on next-generation sequencing technology that allows you to, as the name implies, sequence RNA of a cell or tissue sample. Essentially, this can give you transcriptome sequencing, so it can give you insights into what's going on in a cell at a given time. So essentially, when you sequence DNA, it's a pretty static finding. So DNA taken from different parts of different tissue will actually pretty much yield the same thing, provided it's from the same organism. However, transcriptome sequencing can give you various different insights into what's going on in different tissues at different times, or in different times of an organism's life cycle, etc. And so what you can do here is look for differential gene expression. You can look for post-translational nucleotide variants or gene fusion events. So there's some pretty important implications here. You can actually determine the differential gene expression of healthy tissue versus diseased tissue, or in various disease states. And so you can kind of monitor how things are changing, and then ideally, with precision medicine, you can tailor treatments to the specific tissue and the specific variants that are going on. So for sample preparation, it's relatively simple to describe, although I think in a lab can be a little more challenging. What you do is you sample the RNA from your tissue or cells, transform them into a cDNA library, and then using NGS technology, you sequence that DNA library. So RNA-seq, while it's very powerful, it's actually produces a ton of data. As you can imagine, in a given tissue, there can be a lot of RNA, and so even a small RNA-seq trial will give you a ton of data. And so you need some real bioinformatics oomph to power through the analysis. And one tool set that's been developed is the Tuxedo Suite, developed by researchers at UC Berkeley. So the Tuxedo Suite is essentially just a collection of tools that power through RNA-seq data. Using the Tuxedo Suite, you can determine differential expression, identify splice junctions within the RNAs. You can do short read mapping, identify isoforms, and finally you can visualize the data. So the tools that we're using in this presentation are TopHat, CuffLinks, CuffCompare and CuffMerge, CuffDiff, and then finally for the visualizations, Cummerbund. So the first tool in the Tuxedo Suite is TopHat. TopHat, kind of as the first step, is to align all of your RNA-seq reads, and this uses Bowtie as its engine to do that, so to speak. And so in and of itself, it's actually capable of discovering splice sites, which is pretty impressive, but makes sense when you're looking at the reference genome. So frequently you have to supply your own reference genome to do this. And interestingly enough, the developers of TopHat recently came out with a TopHat 2. This is the second version, and what this does is it actually allows the alignment to go ahead, and you don't have to worry about insertions or deletions or fused genes within your samples.
So from TopHat, we move to the next step in the Tuxedo pipeline, which is CuffLinks. CuffLinks measures global isoform expression by inferring the splicing structure of each gene, but this can be challenging because genes can have multiple alternative splicing. So the tool reports as few full-length transcript fragments as are necessary to explain all the splice variants in the data. And after the assembly, we can estimate the relative abundance of each fragment. So it is determining differential expression and regulation of the genes. CuffMerge is the next step. It merges the assembled transcript fragments from CuffLinks to reveal complete genes, and this is the final step in transcriptome assembly. CuffCompare is an optional step when the when a reference transcriptome is available, it will compare the assembled against the reference and sort out known genes and isoforms from novel finds, which can then be validated with cloning and PCR techniques. CUF-DIF calculates expression in 2 or more samples. It takes the final assembled transcriptome and maps reads to find differential expression. It groups together isoforms of genes that have the same transcription start site and therefore can calculate the total expression of a transcription start site group by adding up the isoform expressions. It will report gene and transcript expression level changes, the p-value, fold change, and the name and location in the genome of the gene and transcript. Cumberbund was created because browsing the CUF-DIF output files by eye can be challenging, and with Cumberbund you can create plots that make it easier to visualize expression levels identified by CUF-DIF. Users can create multiple different plot types, including volcano plots, density plots, heat maps, scatter plots, expression plots, and more. And if they don't want to use any of those, they can even design unique plots and analyses using ggplot, which is the software that Cumberbund is based off of. These plots are ready for publication, and the tool itself transforms all the data from CUF-DIF into an R environment, so the user does not have to do it manually.
For our workflow, we found a sequence read archive for a cohort of patients that were having whole exome sequencing for the detection of copy number and allelic imbalance in chronic myeloid leukemia patients. These sequences were sequenced on an Illumina Genome Analyzer 2X with 76 base pair paired end reads. Unfortunately, this repository ended up having over 370 gigabytes of data files once the archives were unpacked into the FASTQ files, which ended up being far too large to run on the public instance of Galaxy. As a result, we were only able to do the full Tuxedo Suite analysis on one sample. To begin our workflow, we imported the paired end reads for one patient that was found in the data archive. Once this lengthy upload finished, we began using the Tuxedo Suite tools, all of which can be found in the NGS RNA analysis section on the public instance of Galaxy. When using TopHat, we assumed human genome build HG38 for the reference genome. Once the TopHat tool finished aligning the reads, we used the accepted hits BAM file as our input for cufflinks, which then assembled the transcripts. CUF-compare was used to compare the assembled transcripts to the HG38 reference annotations. Due to the immense size of the FASTQ files and the long processing time on the public Galaxy server, CUF-merge was not used in this workflow, as only one dataset was able to be uploaded and analyzed. CUF-DIF can be used to determine differential expression, but I'm not sure how useful this would be with only one patient sample for a dataset. I was hoping the comparison to the HG38 reference annotation would give interesting data. We made sure to use the CuffDiff option to generate a SQLite database for use in visualization with the Cummerbund tool. Default values were used for all tools. Here are our settings for the Top Hat tool. We had to change the input from single end to paired end data near the top, then choose our forward and reverse reads. These were assumed to be forward labeled 1 and reverse labeled 2, as seemed appropriate. And then we selected a reference genome. Galaxy has all sorts of built-in reference genomes, so depending on your data set, choose the appropriate reference genome for your comparison. Once Top Hat finished aligning our reads, we took the output BAM file and used it as the input for the cufflinks tool. We kept all the parameters for this tool on their default. After cufflinks finished processing, we ran cuffcompare on our new dataset using the assembled transcripts from the previous tool. We made sure to use the reference genome HG38 for this tool. Finally, we used the data from the cuffcompare tool and used it in the cuffdiff tool, hopefully comparing our data that was compared to the reference genome along with our original accepted hits to see if there is any differential expression that could be determined in the CML patient from the reference genome. We have made sure to use this Generate SQLite option in order to have a SQLite database for use with the Cummerbund tool. The Cummerbund tool uses R and the data derived from the cuffdiff tool to generate a variety of different plots of your data for publication. This would likely be more useful and visually pleasing given a greater number of patients and data sets that could be analyzed, which was unfortunately not possible due to technical limitations of the public Galaxy server.
Here we have a FPKM density distribution plot. FPKM stands for fragments per kilobase of exon per million reads mapped. I'll try to simplify that. It really means the rate of fragments per base multiplied by a really big number. So that we can try to get some sort of sense as to how often did we see that our reads mapped to the reference. Important things to remember with the FPKM, that it's a relative measurement because it really depends on what genes you're sequencing, what their features are, because different features have different lengths. Depends on your sequencing coverage. So if you sequence billions of reads, you're obviously going to have a larger FPKM than an experiment that only had millions of reads. So it's really a relative measurement that you can use to compare different samples that were run the same, sequenced the same, and sequencing the same genes. So this is showing blue is our first set of reads. We can see that the distribution is much more similar to one another. And it is a normal distribution, while the second set of reads still a normal distribution, but we can see a little bit of noise there on the lower end of the spectrum. So in conclusion, RNA-seq analysis is a very powerful tool for molecular biology. It provides powerful insights into how genes are being expressed at different times. We can use it to find new genes. We can try to figure out why in one person this gene is being overexpressed as compared to a different person. We can really try to figure out what is going on in a live person as compared to the static genetic sequencing. But with all technologies that are based off of next generation sequencing that produces billions and billions of reads and terabytes of data, we need powerful bioinformatics software tools to be able to parse through that data and try to make sense of it. There's always going to be a need for tools to get better and better. Tuxedo Suite has been around since 2009 and has become one of the most widely used because it's very well documented as to its efficiency and accuracy. One thing that's really nice with the Galaxy is it allows for people who are non-computationally minded. They can run through these workflows, because there are many out there that exist, without having to know a whole lot about computers. The Galaxy provides a really nice interface for these biologists to go in and perform their own analyses. So just some pros and cons about the Tuxedo package in general. It's really good about memory. It does not use very much, which is important for speed. And it does have a unique approach that it aligns reads directly to the annotated transcripts, as opposed to a regular genomic reference sequence. We talked about on the other slide where Galaxy allows you to use all these tools without having to know really much about computers. Some of the trade-offs from Tuxedo, because of it aligning reads directly to the annotated transcript, it requires an extremely well annotated reference transcriptome. If you were to do a RNA-seq experiment and you did not have a annotated reference transcriptome, Tuxedo Suite would not be the tool to use. There are other software packages like Trinity that will do that for you as a de novo assembly. And like I said, this came out in 2009, and as always, software improves dramatically with time, so there are much faster and more efficient software packages out there. Trinity is one of them. There's Sailfish is one, Callisto, but those are not as well documented. But just trying to show that tools are always developing, and they are working on TopPath 3 that'll utilize some of that new approaches. Another thing with Galaxy, the larger the data set you have, the longer it's going to take, and especially using that public instance, you're probably better off installing your own local instance or learning how to run command line tools. But overall, the Tuxedo Suite is a really good package. It provides all the insights that you need. All the documentation out there on the internet makes it very easy to use and to troubleshoot if you were ever to run your own RNA-seq analysis. So I would recommend it to whoever, and whenever you're using RNA-seq, if you feel like you've had a good reference transcriptome, Tuxedo Suite is the way to go. All right.